Hello guys, welcome to Gyanvarsha. Today we will be learning about the control of microorganisms. I have divided this topic into two parts. Okay, part 1 is frequently used terms and physical agents in control, and part 2 will be the chemical agents used in the control of microorganisms. So let's start. So the first term is sterilization. Okay, sterilization is the process by which all living organisms, such as all living cells, viable spores, viruses, and viroids, are either destroyed or removed from an object or habitat. Okay, so this means that the main word here is all. Okay, sterilization is the complete removal of all the living organisms or all the living cells from a particular media or whatever thing you want to sterilize. The next term is disinfection. Disinfection is killing or inhibition, removal of organisms that may cause disease. But here the main term is inanimate objects. Okay, it is generally used for inanimate objects such as your tables, chairs. When cleaning or disinfecting your tables or your chairs or such inanimate things, you use the term disinfection. Here there is almost 100% but not 100% removal of organisms. So the term here is disinfection, generally used for inanimate objects. It is used to kill or inhibit the growth of microorganisms on inanimate objects. The next term is sanitization. Sanitization is similar to disinfection. Here, the microbial population is reduced to levels that are considered safe by public health standards. Okay, there is not 100% or around 100% removal of the organisms, but the germs or the microorganisms are reduced to levels which are considered safe by public health standards. The next term is antiseptics. Okay, so the control of microorganisms on living tissues. This is the main term here. Disinfection is for inanimate objects and antiseptic is for living tissue. Okay, living tissues. Antisepsis is the prevention of infection. Sepsis is infection. Antisepsis is prevention of infection. And to prevent this antisepsis and to prevent sepsis, you use antiseptics. Okay, if you understand the term, then you can, if you just go back what is sepsis, what is antisepsis, and what are antiseptics, you can easily know what it is. Okay, these are not as toxic as disinfectants. Dettol. Okay, for example, Dettol, what you use, Dettol is a disinfectant where you use it to clean your clothes sometimes or your tables and chairs, but you also use it on your wounds. Okay, you also use it on your wounds. So it means that it is a disinfectant and also an antiseptic. Here it is being used on inanimate objects, and over here it is being used on living tissue. Next, there are two suffixes, side and static. Okay, that is important to know the difference between these two. Static is stopping. Okay, stopping the growth of organisms. So chemicals that do not kill but prevent the growth, it is called static, such as bacteriostatic and fungistatic. It means they are preventing the growth of bacteria and fungi. It does not mean that it is killing them. It is just stopping the growth or preventing the growth of bacteria, fungi, or any other microorganisms. Similarly, here there is side. So bactericidal, fungicidal, algicidal, viricidal, germicide. It means here it is killing the microorganism. Okay, substances that kill the organisms are called as they use the suffix side. Okay, I hope you understood the difference between side and static. So all these terms we will be frequently using. So just remember the difference between these. The next term, so the next part is, what are the conditions which influence the effectiveness of antimicrobial agent activity? So they are the population size, the population composition, population size of microorganism. Okay, the population composition of the microorganisms against which the antimicrobial agent is being used. Okay, so how? See, consider if you have around 5 organisms and here you have around 50 organisms. So if you're using the same concentration of antimicrobial agent on both, here you will say it is not effective and here you will say it is effective. So the population size of the microorganism matters or on when you're testing how effective the antimicrobial agent is. Similarly, the composition of the area, or here see, if there are 5 organisms but there are bacteria, fungi, viruses, some agents might not be able to kill the viruses, so here it is ineffective. But here if there are only bacteria, the agent, even if there are many, the agent might be able to kill it, so it is effective here. So on what is the composition, where you are using it, where it will be used in future, so those things matter when you are using the when you are testing the effectiveness of the antimicrobial agent. So the population size and the population composition matters here of the microorganisms. Concentration of the antimicrobial agent. Mostly the more concentrated the agent, more the killing of microorganisms. This is then generally, you all might know this, but there are some places where if it is 100%, if the antimicrobial agent composition is 100%, if you're using very concentrated, it won't be effective. For example, if you see in case of ethanol, if we are using 70, we generally use 70% ethanol in our labs, okay, than using 95%. Why so? This is because the 75% ethanol is more effective than 90%. You have to remember this. Why? Because the activity in the former, which is the 70% ethanol, is enhanced because of presence of water. Okay, the water, 70% ethanol is what? It is 70 ml of your ethanol and 30 ml of water. Ethanol alone is not very effective in killing, it's not very effective to kill the organisms, whereas the 70% ethanol is effective. Why? Because the water here helps the antimicrobial agent, it enhances the activity of this ethanol. Okay, and similar, and the other conditions are the duration of exposure, for how much time you are exposing the surface or your material to the antimicrobial agent, and also the temperature. Generally, if you are increasing the temperature at which the chemical agent is acting or the antimicrobial agent is acting, the activity is enhanced. So you can use, if you are using around 50% concentration of your agent at 40 degrees, you can use around 35% of your this 35% of your agent at 50 degrees Celsius. There is I'm just saying is to show you all that if you increase the temperature, you can decrease the concentration of the antimicrobial agent. Okay, and also the local environment, if you are using it along with some other chemical or in some water, so that also will influence the effectiveness of your antimicrobial agent. Next we will see about the physical methods. So okay, the control of microorganisms is in two ways. Okay, there are chemical methods and physical methods. So in this part we will see about the physical methods. We will see this in part 2. Okay, so physical methods physical methods in control of microorganisms. First, what are the different methods? Heat, low temperature, radiation, filtration. Okay, these are the different methods of physical methods used in control of microorganisms. So first is heat. There are two types of heat. One is moist heat and the other is dry heat. By you can just know the difference from these terms, moist and dry. Okay, so moist heat, there are different methods even in moist heat. So first moist heat, uh, first method in moist heat sterilization is boiling. You all know about boiling, heating a heat what you boil water, you boil milk. Okay, so heating to around 100 to 110 degrees, it will kill the vegetative cells but not the endospores. Okay, boiling can kill the vegetative cells of the bacteria, vegetative bacteria, fungi, but it won't kill the endospore. That is, it does not sterilize. It is killing the bacteria but it is not sterilizing. Okay, next is autoclave. Autoclave is nothing but this is an autoclave. It is just like a pressure cooker. Okay, this is the compartment where you all put your things such as flask, media and everything. There is, okay, there is a, here there's a gasket, here there is a coil, heating coil. So, and here you fill it with water. Okay, there is water over here. Okay, there is water. Here there is a coil, your heating coil, and here there is gasket for you all to keep your things. So you all just put your things here, and then you set it, you close it tightly, you check your rubber tube. You then when you start it, you have there, it will run up to a temperature of 121 degrees Celsius. The pressure is 15 lbs pressure, and it will run for 15 minutes. Okay, so it works on the principle of a pressure cooker, similar to pressure cooker, and there will be a complete removal of organisms. Okay, it will be sterile. You will get it is a very good method for generally used in labs. Next is pasteurization. Pasteurization is generally done for milk, wine and other such beverages. Okay, so heating to 50 to 55 degrees Celsius for a few minutes. This is called pasteurization. It does not sterilize but kills the pathogenic microorganisms and slows down spoilage. Pasteurization of milk we have seen, and there is heating to 55 to 60 degrees Celsius. There are two types of pasteurization. One is flash pasteurization or high temperature short term. Okay, from here you can know what it will be. High temperature for short term. Okay, quick heating to 72 degrees Celsius for 15 minutes, then rapidly cooling. High temperature for a short time. Here normal pasteurization is 55 to 60 degrees Celsius for few minutes. Here high temperature short term is 72 degrees Celsius for 15 minutes. Then the next is ultra high temperature sterilization. Here there is heating to 140 to 150 degrees Celsius for 1 to 3 seconds, and this milk does not require refrigeration for up to 2 months. Okay, so what it is, ultra flash pass high temperature short term, high temperature for short term, or ultra high temperature is 140 to 150 degrees Celsius for 1 to 3 seconds. This is about moist heat sterilization. So what are the different things in moist heat sterilization? It is boiling, then autoclave and pasteurization. Pasteurization is of two types, flash pasteurization and ultra high temperature sterilization. Next, dry heat sterilization. There is no water used over here. So this is generally done using an hot air oven. Okay, hot air oven. This is a hot air oven. In this, items are placed here. Here you keep your items here, your flask, okay, your plates, whatever it is, you keep it over here. Items are placed in an oven, and the temperature goes to around 160 to 170 degrees Celsius for about 2 to 3 hours. Okay, here the cells die due to oxidation of cell constituents. Because you are exposing it to continuous high temperature for such long time, There is oxidation of the cell constituents, and also because of such high temperature the proteins get denatured. But here again, the dry heat, it is less effective than the moist heat sterilization. Okay, this is important. So what it is, moist heat sterilization is better than dry heat sterilization. Why? Again, because the water present here helps the heat to penetrate inside the microorganisms and kill them. Hence, the moist heat is the sterilization using moist heat is more effective when compared to that of dry heat. This dry heat sterilization is used for glassware, pipettes, powders, oils, etc. So we generally use it to just dry it off when you're using, generally used in chemical labs. It's not required complete sterilization, then you can use this dry heat sterilization. Next is Next is using of low temperatures. These low temperatures are generally used to inhibit the growth. Instead of destroying the microorganism completely, we just inhibit their growth. Okay, instead of destroying the microorganism, if you put them at low temperature, their growth is inhibited. And this low temperatures, keeping at low temperatures, is nothing but freezing or refrigeration. Generally, refrigeration is at 4 degrees or at 10 degrees, and freezing is about -20 or -80 degrees. So freezing items at -20 or lower temperatures stops the microbial growth because low temperatures and absence of liquid water. All the biological processes which are going on happens in liquid form or in the liquid water. But if there are no water, then the enzymes and the proteins stop functioning. So freezing is a good method for long-term storage. Refrigeration is for short-term storage. This is important. Next, the next type of physical method is radiation, using radiations. There are two types of radiations. One is non-ionizing radiations and the other is ionizing radiations. Non-ionizing radiation, the example is UV light, UV radiation. So the UV radiation of about 260 nanometers is quite lethal, but it does not penetrate glass, dirt films and water. It is lethal, but it does not sterilize. It is not used generally for sterilizing an object, or it is just used for reducing the number or reducing the number of the organism. But what is its mechanism? Okay. So when UV light is absorbed by the microorganisms, what happens is in the DNA there will be formation of thymine dimers. Okay, there are formation of thymine dimers, pyrimidine dimers, and there is formation of this cyclobutane ring. Because of this, there is error in replication, which will lead this error in replication and transcription, due to which the proteins are not functional and the organism dies. Okay, this is the mechanism by which non-ionizing radiations such as UV rays are involved in killing of microorganisms. But this doesn't completely sterilize a particular object or generally used in laminar airflows. It does not sterilize completely. It's just an additional step we use while using a laminar airflow. Next is ionizing radiations. Ionizing radiations such as gamma rays, X-rays and electronic beams, they have good penetration power, hence they are the excellent sterilizing agents. They destroy even the endospores. Even the bacterial endospores are destroyed of both prokaryotic as well as eukaryotic cells. Not only prokaryotic but also eukaryotic cells. But they are not very effective against the viruses. Okay, so they are generally used during this gamma radiations are generally used, that is called as cold sterilization. Cold sterilization means It is not, um, there is no heat involved. Okay, well, what happens is, if because of heating there will be loss of like if the antibiotic or vaccines have particular structure, if we heat it, we sterilize it using some heat, what happens is the structure is destroyed and it will become ineffective. Hence this we need some cold sterilization. So these ionizing radiations are mostly involved in cold sterilization, and they're used to sterilize antibiotics or certain hormones, your sutures which are used in case when you have some wounds, the plastic disposable supplies such as syringes. For to sterilize all this, we use cold sterilization, generally by using gamma rays. Okay, it has been used to sterilize and pasteurize meat and other food. These ionizing radiations are also being used in food industry. The next one is filtration. Filtration is an excellent way to reduce microbial population in case of heat sensitive materials. Hmm. When you have such a some such as your vitamin solution, okay, you have some vitamin solution, sometimes you have a carbohydrate solution, such things you cannot autoclave, because again the same thing, it will destroy, they won't be effective anymore. You're putting your vitamin solution in a culture media so as to give it to your microorganisms to grow, but what happens if you autoclave it, the vitamins are lost completely. So in such cases, when the materials are heat sensitive, you use this filtration, and it's generally used for when your material is in solution form. Instead of destroying the microorganisms, the filter simply removes them. So here is the filter. What you do is, see, if you have a tube and you have your vitamin solution in this tube, you take around 1 ml of your vitamin solution in a syringe. Then you remove the needle of the syringe, attach your syringe over here, and you pass the solution through this filter. The filter is here in this. Okay, the filter is here like this. Okay, this is the filter. There are different pore sizes. Here this filter is about 0.2 micrometer. And when you filter it, here slowly you'll get your filtrate over here. The microorganisms will get trapped in this filter. So there are two types of filters. There are depth filters and there are membrane filter. This is a membrane filter. This is a membrane filter. So generally we use membrane filters in labs. Filters with pore size of zero point 0.2 micrometers are used to remove vegetative cells but not viruses. Viruses can pass through this membrane filters. Okay, about if the filter is about zero point 0.2 micrometers, the bacterial cell, vegetative cells are get filtered, but the viruses are not filtered. They pass through and they will be in your solution. This filters also, air is sterilized by using these filters. Okay, so what are the two examples? Surgical mask, what you use, it is a type of filter where air is being sterilized, and also cotton plugs, which you generally use for during autoclaving or to pack your material, you use cotton plugs. And this is also a method, it is a method to sterilize the air. Okay, laminar airflow consisting of high efficiency particulate air filters, which are HEPA filters, full form is very important, which removes 99.97% of zero point 0.3 micrometer particles. They are the one of the most important air filtration systems. So what are the different air filtration systems? One is your surgical mask, other is your cotton plugs, and the third is your HEPA filters or your laminar airflow. Okay, one are mask, two are cotton plugs, and the third is laminar airflow, which use HEPA filters. So let's study a few things about the laminar airflow. There are two types, horizontal and vertical. They work on a same principle, but the only thing is the direction of airflow. Okay, here this is the blower. It will take up the surrounding air and it will pass it over here. So what happens is all the air get, these are the HEPA filters. The air is filtered through this, and the air blows here. Here the person is working. Okay, here this is the workbench. Person is working here. The direction of airflow is like this, horizontal. And here in vertical laminar airflow, what happens is the surrounding air is sucked in like this. Then the blower gives it to the HEPA filters. The HEPA filter is placed like this. So this is the HEPA filter, and the air flows like this. So here the airflow is vertical. So this directly the person working is more protected over here and compared to that of laminar horizontal laminar airflow chamber. There are different types. This is laminar. There's also biosafety levels. In that also different types of laminar airflows are used. Biosafety level 1, 2, 3. If you all want a lecture on that, I will take it separately. So but for now this is sufficient. So these are the different types of different methods used in different physical methods of sterilization. So what are they? Heat. Heat is using moist heat or dry heat sterilization. Moist heat sterilization generally done using an autoclave, which works on the principle of a pressure cooker. Here, the important thing is the temperature is 121 degrees Celsius. The temperature is raised to 121 degrees Celsius. The pressure is raised to 15 lbs, and at this particular condition it is sterilized for about 15 minutes. Then the thing is said to be autoclaved or sterilized, almost sterilized. And next, dry heat is generally done using an hot air oven, where the material is placed at 160 to 170 degrees Celsius for 2 to 3 hours, generally used for powders or your glassware. And moist heat sterilization is more effective when compared to dry heat sterilization. Lower temperature is generally used to stop the growth of microorganisms. It is also a good method for storing your microorganisms. You store your glycerol stocks at -20, -80, -80 degrees Celsius. Why? Because there is no water, there is no activity in the organism. The growth completely stops. So if you can just take out the glycerol stalks, thaw it, and you can just use it. You can streak it and you revive your organism like this. It is good for storing a microorganism in the form of slants or glycerol stock. Okay. Next, the next is radiation. There are two types: non-ionizing and ionizing radiations. The ionizing radiations are used in food companies and for pharmaceutical companies, cold sterilization, etc. Next, there is filtration, generally a good method for when you have a solution to sterilize and which are heat sensitive. So there are two: membrane filters and there are depth filters. So you can also filtration is also used not only for solution but also can be used for air. This air can be sterilized using your surgical mask. Your cotton plugs are a type of filters, masks are a type of filters, and the other is laminar airflow. The laminar airflow is nothing but uses HEPA filters, which are high efficiency particulate air filters. Your laminar airflow are of two types, horizontal and vertical laminar airflows. So this was all about your physical methods of sterilization. In the next part, we will be learning about chemical methods of sterilization. Let's see a few questions before that. So the first question is, which one of the following statement about control of microbial growth is not correct? Okay, statement about microbial growth is not correct. This question is from GATE 2020. So let's see the options. Non-ionizing radiation leads to thymine dimers formed in DNA. I told you, right? Non-ionizing radiation, what was the example we studied? It was UV rays. So they form thymine dimers or pyrimidine dimers, and also there is formation of the cyclobutane ring, due to which the microbial growth is stopped. So this is correct. Next, spirochetes and mycoplasma can pass through membrane filters. This is also correct. Okay. Then use of high concentration of salts and sugars to preserve food is a chemical method of microbial control. Are salts and sugars chemicals? No, right? So salts and sugars are not chemicals. So from this you can easily eliminate this option that they are not chemical method. Using salts and sugars are a method of preservation, but it is not a chemical method of preservation. And thermoduric bacteria can survive pasteurization. Thermoduric is which can survive high temperature. Thermo means temperature, and duric is which can survive high temperature. So thermoduric bacteria can survive pasteurization. This option is also correct. So here the wrong option is, so the wrong statement is, use of high concentration of salts and sugars to prevent food is a chemical method of microbial control. The next question, which of the following radiation makes methods is used in preserving food from spoilage by microorganism? Which of the following radiation methods is used in preserving food from spoilage? Which methods have we studied? Following radiation methods is used in preserving food from spoilage by microorganisms. I told you in food industry they generally use which rays? Gamma rays. So gamma rays are a type of ionizing radiations. So the correct option here is ionizing radiation. Okay. Next question. Which of the following is not an accepted method for sterilization? Okay. Autoclaving, I told you it is a good method of sterilization. Autoclaving is a good method of sterilization. Gamma rays are also a good method of sterilization. X-rays are similar to gamma rays, which are ionizing radiation, which is also a good method of sterilization. So the one remaining here is UV rays. UV rays do not sterilize, they just can eliminate a few organisms. Okay, it is not an accepted method for sterilization. So the correct answer here is UV rays. This has come in 2018. Next question. This is a type of little bit of application. Okay, a bactericidal agent. Cidal. What is cidal? Cidal is killing. Okay. Agent X is added after 3 hours of growth of a bacterial culture. What does this sentence mean? After the bacterial culture was growing for 3 hours, then the killing agent was added. Okay, killing agent was added after 3 hours. Okay, so following the addition of X, the bacterial growth was measured using the standard plate count till 24 hours. Which of the following figures is the most accurate representation of the action of X? So let's see. In this, what is the x-axis is time, and the y-axis is number of bacterial cells. Okay, here it is showing that 3 hours bacteria is growing, but after after the addition, here there was addition of the agent. Here there was addition of agent, then again it is still increasing till 24 hours, which is not what the agent does. So this is wrong. Next option, here we can see first what they said, that up to 3 hours the bacterial culture was grown. So where is the 3 hour? This is the third hour. Okay, then over here the agent was added. As soon as the agent was added, you can see decrease in the growth. Okay, the decrease in the growth. So this might be correct. Next, let's see the other options. So here again, after adding the agent, still the bacteria is continuing to grow, which means this is not correct. And over here it shows that the growth has stopped, but we are not stopping the growth, we are adding a cidal agent, which means it will kill the bacteria. So the graph should have gone down. So this is also wrong. The correct option is B. and this has come in GATE 2017. One more question. Of the following, the most effective method to kill bacterial endospores? Which is the most effective to kill bacterial endospores? It is moist heat sterilization. Okay, moist heat sterilization, autoclaving. Okay, it is the most effective and for generally used in research labs for killing the bacteria, killing the organisms, vegetative cells as well as endospores. This question has come in GATE 2015. So that's it for today. Thank you. If you all want any other topic, please let me know in the comments. The next video will be of the chemical agents used in growth control of microbial control of growth of microorganisms.